with you here today. This week in our new segment, #Trending, we are investigating the origins of the internet's latest craze, #DabForDevils, which has been trending globally for the last 3 weeks. Crossing live to Ron Burgundy with more on this topic.
G'day Sally. #DabForDevils is a non-profit organisation acting to raise awareness of devil tumour disease, otherwise known as DFTD. The Tasmanian devil is a small black carnivorous marsupial. Found in Tasmania, large males can weigh up to 12 kilograms. However, there is a large variation of body sizes among the species. According to fossil records, 400 years ago, the Tasmanian devil had a wide distribution, spanning Australia and Tasmania. As a result of predation and more recently the emergence of the devil facial tumour disease, the populations had a significant decrease in numbers and genetic diversity, and now have a greatly restricted geographical distribution. Tasmanian devil tumour disease is a transmissible cancer, and tumours spread progressively through the oral cavity, jaw and face. Affected individuals die from a combination of starvation and secondary infections within 6 months of contraction, and there has been no evidence of any individuals recovering. DFTD is transmitted from animal to animal by cell implantation during fighting as a result of bites of another individual. The first case of DFT was thought to have originated in Tasmania in 1997 from a female devil. It has since then spread to affect the majority of the species' geographical range, with up to 90% of individuals within certain locations being affected. DFTD is posing a huge threat to the survival of the species, and current projections threaten the Tasmanian devil with extinction within the next 25 years. Understanding the genetic evolution of the DFTD is an essential aspect of developing solutions such as vaccines and using epigenetically modified DFTD cells. I am now here with some devil trappers to explain to you what we're doing here today.
I am Sheila Jones. And this is Jill Jack. Today we'll be discussing with you some of the genetic factors of the DFTD. Research has shown that DFTD, as both a pathogen and an allograft, are unaffected by the host's immune system. The devil's immune system fails to reject the DFTD cells, resulting in a highly contagious disease with 100% mortality post-contraction. This immune evasion is only displayed in one other naturally occurring contagious cancer, canine transmissible venereal tumour, or CTVT. CTVT is an STI tumour which has had research conducted into its immune evasion, so the comparison between the diseases can provide an insight into the relatively unresearched methodology of DFTD. DFTD cells do not express the surface major histocompatible complex MHC molecules due to low genetic diversity and downregulation of genes that are essential to the antigen processing pathway. MHC molecules are the surface proteins that allow recognition by the immune system in vertebrates. They determine the histocompatibility and allow recognition of foreign and potentially harmful molecules. This loss of vital gene expression is a result of regulatory changes such as epigenetic deacceleration of histones, which is a vital part of gene regulation reactions. Without the downregulated histones, the MHC class 1 that are produced by the DFTD cells are retained in the endoplasmic reticulum and processed. T cells, a type of lymphocyte that contain T cell surface receptors, are required for cell-mediated immunity. These T cells do not target the DFTD cells due to the poor recognition, as they have lost their MHC molecules. This provides a basic insight into how the immune evasion occurs in DFTD. CTVT also uses downregulation of MHC class 1 expression during transmission of cells and tumour growth, bypassing the immune system and establishing a population. It then uses upregulation of expression as it infiltrates the lymphocytes and releases interferon gamma IFNY. IFNY is a type 2 infro with immune functions. It's critical for immunity development against viral, bacterial and protozoal infections and an activator of class 2 MHC molecules expression. CTVT uses these methods to establish and remain in the host without immune responses. Both diseases show a distinct ability to adjust MHC expression resulting from a different cellular environment.
The pathology of devil facial tumour was documented in an experiment carried out by our Animal Health Laboratory in Tasmania. The results determined that the masses were typically firm with flattened ulcerated sections and majority of the tumour were being less than 3 centimetres in size. The neoplasms, which are tissue samples, only varied slightly in colour, and some being more translucent than others. All the samples appeared relatively similar when viewed microscopically. The tumours typically originate in the dermis or submucosal connective tissue within the oral cavity and form highly vascularized aggregates. The majority of the samples, 75%, showed some necrosis, tissue death, and in 65% of the samples, tumours had metastasized to other areas of the body, and of those 65% of cases, 57% of samples had metastasized to the lymphatic system. Other areas of the body that had visible tumour growth included the lungs, heart, and spleen. The microscopic analysis of neoplasms in this research once again confirmed that devil facial tumours are malignant, aggressive, and poorly differentiated. It is because of this aneuploidia, poor cell differentiation, and the fact that most individual cells had minimal organelle presence that the DFTD cells are considered to have marginal specialised functionality.
Several cracks here, as you can see, Jill Jack. There's some droppings too. Still fresh, but they're close.
There's actual poop here.
No thanks.
Thanks so much, guys. Now we have some live footage of devils and crossing over to Brick Tamland in rural Tasmania.
Look, it's a Jesse devil!
Yes, that's right, Ron. Here I am in sunny Binalong Bay with the real things themselves. I've just been informed of some groundbreaking genetic research. Due to down-regulation of the genes and transporters essential for antigen processing pathways, have resulted in the DFTD cells to not express any MHC cells on the cell surface. MHC molecules can be restored to the surface of the cells by using IFNY, a double recombinant, which is involved in the upregulation of MHC2 transactivator. It is proposed that MHC positive or epigenetically modifying DFT cells may provide a vaccine to the devil population. Some human immunotherapy strategies activate antibodies or mitogens with non-specific stimulation of cytokines in order to induce MHC1 killing of tumour cells. Due to the lack of MHC expression on DFTD tumours, cytotoxic T lymphocytes are not able to target the tumours, explaining the poor response in vaccine trials. K562 tumour cells used in xenogenic immunisations lack surface MHC1, but are recognised in the Tasmanian devil's body. Natural killers, like cells in the devils, can consistently kill K562 cells by antibody-dependent cell-mediated toxicity, otherwise known as ADCC. Although Tasmanian devils can form strong ADCC responses against MHC negative cells, antibody responses against DFTD are poor. A technique that can consistently activate the Tasmanian devil lymphocytes to kill DFTD cells in vitro can be achieved using cytokine-rich supernatant from con-A cultures. A recombinant Tasmanian devil IL-2 and the toll-like receptor against poly-1C, a promising result for immunotherapy of the DFTD. And that's all from me. Back to you in the studio.
Thank you, Brick. The future does not look bright for the little Tasmanian devil. Genetic intervention, whether through evolution or human engineering, will need to occur for this endangered species to survive and thrive as they did in the past. Well, that's all we have here for BBC News. We'll see you next time, and remember, Dab for Devils. We're sending you out with some exclusive footage of Dab for Devils. See you later.